Thank you very much for inviting me.
So I'd like to start off with an observation that in American slang, if you call somebody a vegetable, what you're saying is that that person is not moving, is paralyzed, and more than that, they're likely not conscious. So you might veg out, become a couch potato, and all of those things sort of imply that plants neither move nor are able to sense their environments. And I'd like to really pose that question. Are plants capable of sensing their environments? And I hope by the end of the talk that I convince you that in fact they are, and that they respond to all sorts of environmental cues. I don't know exactly why this idea has been very contentious. We've known for a long time that plants are very sensitive to their light environments. So if any of you have grown houseplants and you put them near the window, they will very quickly grow towards the source of light. If you grow plants out in the open like this, they won't get very tall, but they'll often get bushier. If you restrict the amount of light that a plant is receiving, instead of getting bushy, it grows tall, it heads for the light. So clearly plants are able to perceive the light that is reaching them. And we've now found, for instance, that plants can differentiate when they are shaded between being shaded by an inanimate object and being shaded by another plant. And they respond much more strongly to being shaded by a plant than they do to an inanimate object. Plants are also able to use chemical cues, volatile cues, such as those that Junji was talking about in the previous talk. This is an example of a daughter plant, a parasitic plant, and that's this thing here. And it is able to perceive the volatiles that are being emitted by a good host, in this case, the tomato plant there on the right. And so it orients towards the host plant. So it's using volatile cues to find its host. In addition, plant roots are able to sense the quality of nutrients that they are growing through in the soil. These are the root systems of two barley plants. For the plant on the left here, the nutrients were present throughout the medium that the plant was growing in, and so the roots are pretty evenly dispersed. For this barley plant on the right, the nutrients were concentrated in that band near the top there, and so the plant proliferates roots only in those regions that have the nutrients, and it grows very quickly through those regions that are that don't have the nutrients. So plants are able to perceive and respond to the chemicals in their environment. We also know that plants are attacked by a lot of insects, a lot of herbivorous insects such as these caterpillars. And I'll spend the rest of this talk considering whether or not plants can perceive the cues of these potentially very risky organisms, and do they respond to these cues? We've known for some time that when plants are attacked, they change, and some of those changes allow them to be better protected against the things that might attack them. And some of these changes are chemical changes, such as nicotine on the left. Some of the changes are physical, such as these hairs that are indicated on the right. So plants are increasing their defenses in response to getting attacked. In addition, again, I will hope to convince you in this talk that not only do plants respond to getting attacked themselves, but they also respond when their neighbors have been attacked. So before they themselves have been colonized or attacked by these insects, they can begin to increase their defenses. So what evidence do I have to support this claim? I have been working in this system involving sagebrush, very slow-growing, very common shrub in the western part of North America. And this is the basic experiment that I've been doing, that I've now done many times with sagebrush. And in these experiments, I have 3 different treatments shown by these 3 cartoons. Here at the bottom, this is a control. I don't do anything to this control bush. And at the end of the season, I count up how much damage this assay branch has received. So how much damage by herbivores does this branch get? Up in the upper left, I damage a neighboring branch with scissors, and I record how much damage the assay branch on the neighboring plant. So the neighboring plant that has not been messed with, how much damage does the assay branch there receive? And then in the third treatment, I experimentally clip a branch on the neighboring plant, but then I immediately cover that clipped branch with a plastic bag. And the plastic bag minimizes the possibility of volatiles moving between the clipped branch and the assay branch. And as I say, we've now done this experiment a number of times, and much of this work and much of the work that I'll describe through the rest of my talk has been done in collaboration with Kaori Shiojiri, who will give the next talk. And what we have found fairly consistently is that if we look at the amount of damage over the season in the field to the assay branch, it's reduced when the neighbor is experimentally clipped. So the amount of damage received by the assay branch is reduced when the neighbor is clipped relative to the control, or relative to the case where we've blocked the possibility of, or we've minimized the possibility of volatiles moving between the clipped branch and the assay branch, this clipped and bag treatment. And this suggests that a volatile cue is required. We've also done these experiments for potted plants, where there's no soil contact or root contact between these plants. And as long as we allow volatiles to move between the plants, we can see this increase in resistance or a reduction in damage associated with these treatments. So we're pretty confident that the communication that is occurring here is occurring through some volatile chemical moving between the clipped branch and the neighboring assay branch. As I've said, we've done these experiments many times, and in the field. Sagebrush is the most abundant plant in this biome, in the Great Basin of North America, so that sagebrush plants are often neighbors. We can find evidence for communication between plants that are up to 60 centimeters apart. Beyond this distance, we don't see evidence for communication, and it appears to be pretty much of a threshold. It's not stronger as the plants are closer together, and at 60 centimeters, we lose the signal completely. We found that young plants tend to be both better emitters of cue and also more receptive to cues. In the experiments, in the cartoon that I showed you, the initial experimental clipping was done with scissors. We've also repeated these experiments using real herbivores, real insects, to do the damage, and we find very similar results. Okay, what I'd like to ask you to consider next are the fitness, really benefits and costs of responding to these cues of damage. And let's start with seedlings. This is a long-lived plant. In most years, it does not recruit, it does not produce seedlings. But in years that are very wet, and especially in years where it continues to rain or snow late in the season, those are the years that we see new seedlings. And this is kind of, in those years, what it looks like. The ground is a carpet of these little seedlings. So we're able to follow the seedlings, the survival of seedlings, next to bushes that we've either experimentally clipped or that we haven't clipped. Most of the seedlings in both of these years died, but what you can see is that in both of the years, survival of the seedlings was about an order of magnitude greater if they were close to a clipped adult neighbor relative to an unclipped adult neighbor. So it seems as if there's some benefit for the seedlings. In addition, if we keep track of established plants, what we have found is that both the number of new branches that are produced and the number of inflorescences of flowers that are produced is greater for branches that are near clipped neighbors compared to branches near control neighbors. So as well as we can measure, there seems to be a benefit of being near a clipped neighbor and getting these cues, an early warning of a high risk of damage. So plants may benefit by eavesdropping on their neighbors, by being good listeners, and paying attention to reliable information in the environment. If it's beneficial, if this information benefits neighbors, and presumably plants are competing with one another, it raises the question then, why do plants emit these volatile cues that benefit their neighbors? And we've considered a number of hypotheses. Let me go through these first three really quickly. It's possible that these cues just leak out, that they're not adaptive, that the plants are not trying to provide this information to their neighbors. When we get a cut, we produce this very bright red cue that our neighbors are able to recognize, but it would be silly to make the argument that we're producing this very conspicuous cue in order to communicate with our neighbors. We're producing this conspicuous cue for other reasons. We use hemoglobin to move oxygen around. And this is a consequence of other physiological constraints. And it might well be that these volatile chemicals that are produced when plants are wounded are not an adaptation, but are the consequence of other sorts of physiological constraints. And I don't really know how to address that hypothesis, but it seems like one that we ought to keep in mind. A second possibility is that these cues may repel herbivores, and I have no written down because this has, I've not seen evidence of this. In the last year or so, I've started to do a lot of work with a new herbivore, a beetle species, and in fact, it is repelled by these volatiles. So this can occur some of the time. Junji just described cues that attract parasitoid wasps, and there are many systems for which this has been carefully documented. In our sagebrush system, we have not found evidence that these cues are attractive to predators or parasitoids. Another possibility is that these volatile cues may allow plants to coordinate their own systemic defenses. When we get an infection on one part of our body, one finger, we produce white blood cells. These white blood cells move through our vascular system, and we are better defended against that infection systemically. And plants can do this to some extent as well, but they are not very good at it. The vascular connections between plants are not always active, especially for plants that are growing in arid environments, such as the sagebrush that we've worked on. And so plants are not particularly good at coordinating their resistance systems using vascular means. They do a better job of this using volatiles. And let me describe this experiment to you. It's a very similar design to the one involving communication between neighbors. In this case, I'm setting this up just using individual plants. So once again, we've measured the amount of damage over a season on an assay branch. And this is a control, so nothing was done to this particular plant. We've got a clip treatment where we've experimentally clipped the leaves of one branch, and then we measured the damage on the assay branch. So here there's the possibility for vascular communication between the clipped branch and the assay branch, because they're connected. And there's also the possibility of volatile communication between the two. We have a clipped and bag treatment where immediately after clipping that branch with scissors, we've enclosed it in a plastic bag, which allows vascular communication between the two, but minimizes volatile communication, since the plastic bag prevents the volatiles from moving between the branches. And as you can see, only the clip treatment had reduced levels of damage to the assay branch. So only where the volatile communication was possible was the plant able to mount a systemic, a whole plant defense against herbivores. Where the vascular communication was possible, damage to the assay branch was pretty high, was as high as the control. So one advantage to emitting these volatile chemicals may be that it allows the plant to coordinate its own internal defenses. This got us thinking that if much of what the plant is doing is perhaps just communicating with other branches on the same individual, maybe there's some kind of kin selection going on. So these plants have been found to be highly genetically structured. What that means is that individuals growing next to one another are far more related than individuals that are growing at some distance from one another. So neighbors share many genes with one another. And since this communication only seems to spread 60 centimeters, most of the tissues that are going to be responding to the volatile cues are either self tissues or the tissues of the closest neighbor. And so we wanted to investigate this possibility a little bit further. And we did this by making cuttings or clones of plants out in the field. So we've got plants that are rooted out in the field. We make clones of those, take them back to the greenhouse in Davis, grow them up, and then we've brought them back out to the field and put them near either the same individual that we took the cutting from, and we call this self, or a different individual, we call that non-self. And we're interested in comparing how well they communicate with tissues that are genetically identical versus tissues that are genetically different. And we also added a couple other treatments. We added a control where we didn't do anything to this plant. And we also added another treatment where we actually clipped the assay plant. And I should have told you, I know this is the most confusing of the experiments I'll describe, that in every case, the plant that stayed in the ground is the assay plant, is the plant that we measured at the end of the season. And we used the potted plants, whether they are genetically similar or genetically different, just as a source of information, a source of cue. Okay, here's what we found when we did that. We have, again, the assay plants and how damaged they are on the y-axis. Unclipped controls had a lot of damage. When we actually damaged the assay plant itself, when we actually clipped the assay plant itself, damage was reduced. When we clipped the genetically identical self plant, damage was also quite reduced and was different, was lower than when we clipped the non-self plant that was genetically different. So plants, and this is really the most interesting of the comparisons, plants that are receiving cues from tissue that's genetically identical to themselves respond more effectively than when they are receiving cues from an individual that is genetically different. And this provides the possibility of kin selection. And so we got thinking about this some more, and we wondered whether plants might be able to communicate more effectively with individuals that were more closely related. but there are all these plants out in the meadow. How do you know which ones are more closely related to which other ones? And so we were able to enlist the help of Satomi Ishizaki, who's now at Niigata University, and she developed a system of microsatellites that allowed us to estimate the genetic relatedness between individual plants. And so we started moving around the volatiles between plants of different relatedness. And we've done a series of different experimental setups to evaluate this. I'll just kind of describe one to you where we've moved volatiles from a distant relative or volatiles from a close relative. And what you can see is that communication was more effective when the cues came from a close relative of the assay branch compared to a distant relative. So plants are communicating more effectively when the cues are from close relatives, relative or compared to when the cues are from strangers. Again, thinking some more about this, and one of the things that we've tried to do is to see what animal behaviorists and people who study animal communication have been up to, because they've been thinking about similar problems for decades. And one of the things that the animal communication workers have found is that there are geographic dialects. So this is a song sparrow, and song sparrows are found along the Pacific coast of North America. I'm sorry, this is a white-crowned sparrow, not a song sparrow. And these are the sonograms of the different populations. And this is just a sort of representation of what their sounds, what their calls are like. And what the ornithologists have found is that the white-crowned sparrows respond more strongly to the songs of their local populations. With birds, you're able to record the sounds and then play them back. And the birds responded most strongly to their local dialects. And so this seemed kind of cool, and it made us wonder if there were local dialects, essentially, in our sagebrush system. And so we were able to move plants around, and more recently we have figured out that we can leave the plants where they are and just move the volatiles around in balloons, essentially. We have found, with a smaller sample size and less elegantly than the bird people can do, but we have found that plants respond most effectively to cues that are local cues relative to cues that are foreign cues. So there's some evidence of geographic dialects in this plant communication as well. Okay, the thing that I've been excited about most recently, again, sort of trying to steal the low-hanging fruit from animal behaviorists, is the possibility of personalities. So recently there has been a lot of interest in the possibility of animal personalities. And what people are referring to when they use this term are consistent differences among individuals in behaviors. So individual differences in behavior that are consistent across time and ecological context. In other words, Are there some individuals, for instance, if we consider alarm behaviors, are there some individual prairie dogs that are kind of frightened of everything, that will respond very quickly to the alarm calls of their neighbors? And perhaps there are other individuals that are much less responsive, that they're the daredevils. They don't really care. Similarly, are there some individuals that everybody pays attention, all the other prairie dogs pay attention to the calls of that individual. That individual is particularly reliable. And then perhaps certain individuals who might cry wolf. And it doesn't matter if that individual, or it matters less if that individual gives the alarm call. And people have found these kinds of consistent differences among individuals of things like prairie dogs. And so the last couple of years, we have been very interested in seeing whether there are these sagebrush personalities. Do some individual bushes consistently respond differently than others, either as emitters of cues or responders of cues? And I won't show you the data, in part because I don't really know how to analyze it, and so I haven't done that yet. But it does, the impression that we get is that it seems quite clear that there are some individuals that are consistently, year in and year out, better at responding to their neighbors, or more likely to respond to their neighbors than other individuals. And similarly, that there are consistent differences in the effectiveness of the alarms of certain individuals. And this is something that I am interested in pursuing in the next year or so. So in conclusion, I hope I have convinced you that plants are in fact not these unconscious, unresponsive blobs that just kind of sit out there in the environment, but in fact, plants are quite sensitive to environmental information, and that they respond when they perceive this information. I've been especially interested about plant responses to herbivory, and again, I hope I've convinced you that plants are able to perceive volatile cues of their neighbors about their risk of being attacked. So communication happens, but it's not random. Communication is more effective among self or genetically identical tissues and among kin. And finally, there seems to be evidence that individuals have consistent personalities. And with that, I thank you for listening, and I'll be glad to answer questions if there's time.